Today, we're very pleased to host at the Sugar Science this discussion, autoantibodies in type 1 diabetes, biomarkers or mediators of destruction. We have an esteemed panel, Ake Lernmark from a PhD from the University of Lund, Sweden, Dr. Eddie James, PhD from Benaroya Institute in Seattle, Dr. Chris Hampe, PhD, and Ian Sweet, PhD from University of Washington, and Dr. Sally Kent, PhD from University of Massachusetts, the Chan Medical School. Just a little bit about the discussion from recent observations that the group has made, as well as from studies of many autoimmune diseases. Autoantibodies indeed can penetrate into cells and have functional impact. In addition, the robust correlation between the appearance of autoantibodies and the onset of type 1 diabetes suggests a causative row. This message needs to be brought out, and dogma that autoantibodies do not have a pathogenic impact on loss of beta cell function needs to be challenged. Data will be presented and evaluated that supports the validity of this statement and will be discussed with respect to what needs to be done to further develop our understanding of the pathogenic role of autoantibodies in type 1 diabetes. The first presentation by Ake Lernmark will be the historical perspective of the discovery and characterization of autoantibodies during development of type 1 diabetes, followed by a short presentation by Dr. Eddie James, traditional views of the development of T1D focusing on the role of immune cells. Presentation 3 by Chris Hampe and Ian Sweet will discuss the plausibility of autoantibodies to play a pathogenic role in the impairment of insulin secretion in the onset of T1D. And finally, presentation 4 by Dr. Sally Kent, what is needed to show pathological impact and relevance of autoantibodies in the landscape of T1D. Welcome, everyone. This is a very large crowd here, so please mute yourself and feel free to drop questions in the chat, which will be answered in the due course. Thank you very much. Okay, do you want to begin speaking?
I'm all set to begin, and I want to thank Ian and Chris for raising the point of, and Monica for setting up this discussion, which is, I think, very timely based on novel data. So I'm at the Lund University Clinical Research Center in Malmö in Sweden. And if we move to the next slide, and Ian is going to do that. This is what I'm going to talk about the next 8 minutes or so. Discovery of islet autoantibodies and the correlation of the islet autoantibodies with disease pathogenesis, the natural history of these autoantibodies, and why islet autoantibodies were not or are not believed to have a pathogenic role. The discovery based pretty much on the paper or 2 papers in the Lancet in 1974, where indirect immunofluorescence was used to finally detect the anticipated islet cell antibodies that, in comparison with thyroid disease, with Addison's disease, indirect immunofluorescence had been used for several years, but it was technical development, if you go back, technical development that allowed 2 parallel laboratories 1 month apart to publish the first islet cell antibody tests. From my own perspective, I was working on beta cells, trying to identify the glucose receptor or the glucose transporter. Remember, we're talking 50 years ago, and nothing was known how beta cells were able to measure glucose concentrations. And I went on to Chicago, where I did my postdoc and later a junior faculty. And during that time, we were able to use the single cell suspensions that I had developed at the University of Umeå in Sweden. And we found that patients, all of them were from the US by a pediatrician in Missouri who provided the serum. And we could demonstrate that there was a significant number of children who had antibodies binding to the surface of the beta cells or islet cells. And that opened up the question, since I was in the biochemistry department in Chicago, it wasn't the antibodies that were interesting, it was really the antigen. What is it in the beta cell membrane that these antibodies are reacting with? and I was recruited to Denmark to the Steno Memorial Hospital laboratory that became the Hagedorn Research Lab. And I was able to recruit a fantastic postdoc, Steinen Bekkeskov, who had already worked with antigens in infectious diseases in Africa. And we were able to convince ourselves and the world that the sera immunoprecipitated a 64K protein. And it took us 10 years in 4 different laboratories before it was shown by Steinen that the 64K antigen had glutamic acid decarboxylase activity in a very elegant paper in Nature. And in our own laboratory in Seattle, we were then cloning this GAD in human islets. And to our surprise, it was a hitherto not known GAD65 isoform. And in our lab, we also developed a novel assay, which is still used today by in vitro transcription translation of the cDNA to make radioactive autoantigens. So that became the standard for islet cell antibodies, and it resulted eventually in Chris Hampe to be able to clone monoclonal antibodies to the GAD65 antigens. The next slide, please. So the question was at the time when we saw these islet cell surface antibodies, and these are published papers, that just taking the immunoglobulin fraction of sera from diabetic patients were able to inhibit insulin release. And we did a passive transfer experiment in mice, where mice were injected for 2 weeks and found by pancreas perfusion to have a reduced insulin response to glucose. And the other papers were in parallel or slightly thereafter to demonstrate the fact that these antibodies mediate cytotoxic phenomena, either complement mediated or cellular mediated effects. The next slide, please. And these are the now certified biomarkers that can be used to design clinical trials, 4 of them, and they have been approved as enrichment markers by the European FDA. And we assume that the development will continue. The next slide, please. And they are analyzed in a number of studies. This is a consortium of several pre-TEDdy investigations where you can follow the individuals who developed up in the left corner predominantly multiple antibodies at first, and then you follow them until diabetes, or they develop predominantly insulin autoantibodies or GADA first. And the next slide, and you can see that they form 3 different trajectories. The green is multiple antibodies when first investigated at birth or close to birth, and they move very rapidly on to clinical onset. And the curve in the middle is GADA, is IAA, and the upper curve is GADA as the first autoantibody. The next slide, please. So when you look at the natural history, the TEDdy study has shown that predominantly IAA first, the yellow curve to the left, come first, followed by a different pattern for GADA, the blue curve, or multiple antibodies as the first antibody. And you can see the incidence rate here that the IAA first is predominantly very young children. GADA continues throughout. The panel to your right is those children in TEDdy who only develop 1 antibody. So the appearance of the antibodies for us in the TEDdy study came a little bit earlier than anticipated. The next slide. And then there is a number of issues that autoantibodies are not believed to have pathogenic role that has influenced the progress in research. You know, insulitis was described already in 1901, and later on by GEPS, primarily was shown to be T cell infiltration or lymphocyte infiltration, but there were also B cells and macrophages. Nobody could demonstrate the position of immune complexes, and therefore antibodies were dismissed. The islet cytoplasmic antigens or intracellular antibodies were dismissed, and there was a pretty loud experimental animal mafia arguing for T cells, and the antibodies were dismissed. So I think there is a missing link here, and that is to look at very carefully the way T cells and B cells interact to explain the strong ability to predict clinical onset. And we don't have a T cell assay doing the staging that is shown in the next slide, which is that stage one is that a person has developed 2 or more autoantibodies, still normoglycemic, no symptoms, and no insulitis. Stage 2, the dysglycemia appears, still 2 or more antibodies, and that is during stage 2 that insulitis seems to appear, and stage 3 is diabetes. So there is a need for studying these different stages in relation to the function of T cells and the function of autoantibodies. The next slide. Thank you. Yep.
Yes. Okay. All right, that was fantastic. I'm going to get the next slide set.
Okay, so I'm Eddie James from the Benny Institute, and I've been tasked with kind of presenting what I'll call the classic view of type 1 diabetes progression and etiology. And I'll begin, close to where Oka ended. And that's, with this classical Eisenbarth diagram that maps out basically how type 1 diabetes has been observed to happen. We all know that there's a strong element of genetic predisposition. We all know that there is a period of insulitis and basal injury, followed by a period where there's, measurable glucose intolerance and finally clinical onset of disease. And it's believed that, very early in that process, and most diagrams place, T cell autoimmunity even prior to the formation of autoantibodies. And I think immunologically that's appropriate because when we actually, characterize the autoantibodies that we see coming up to antigens like GAD and IA2 and insulin and ZNT8, by the time we're able to measure them, those antibodies are, clonally diverse. They show evidence of affinity maturation. And so they have all the classic traits and hallmarks of a T cell help, type of antibody response. I'll go ahead and go to the next slide. And of course, T cells are strongly implicated in islet damage. There's a strong association between HLA risk and type 1 diabetes development, which I'll go into a little bit more in the next slides. And importantly, antigen-specific T cells have been shown to infiltrate the islets not only of NOD mice, but also of humans. And I have here at the bottom of the slides, 2 landmark studies, one by Copiators et al. From JXMed, and one from Wilcox et al. From Clinical Experimental Immunology, both showing that you can label with HLA class 1 tetramers, CD8 positive T cells, that see antigens like IGRP and IA2. And so, these T cells are making it to the scene of the crime. And they are obviously doing something. And importantly, T cells have been shown to be absolutely required in the NOD model of type 1 diabetes, which is universally held to be an accurate model of the disease with genetics and patterns of disease that closely parallel what we see in humans. This is one of many papers that I could have chosen. And you can see here just from the title of the paper, type 1 diabetes development requires both CD4 positive and CD8 positive T cells. And I've put down here at the bottom of the slide, one of their best figures. In panel D here, which I've labeled CD4, what we in all 3 panels we have insulin stained in red, and then the immune population of interest stained in green. And we can see here in panel D that CD4 positive T cells in these very early stage NOD mice have infiltrates of CD4 positive T cells. Moving on to panel E, we see that they're being infiltrated by CD8 T cells. Panel F, they're recruiting CD11B positive antigen presenting cells. And these mice go on to get diabetes. And this work and others show that basically this is all an adoptive transfer model into a non-skid mouse. And so, CD4 positive T cells by themselves, CD8 positive T cells by themselves, or even more potently, both in combination, are sufficient, to drive disease, and their deletion blocks disease. And so you can make an argument, at least in the NOD model, that T cells are, both necessary and sufficient for type 1 diabetes like disease to happen. In humans, susceptible HLA class 2s are the dominant genetic factor in the disease, and there are many different versions of the article that show many different Manhattan plots. But what all those Manhattan plots have in common is that both the earliest discovered genes, shown by their dark blue color, and the highest odd ratios are always HLA class 2. And of course, functionally speaking, we know that what HLA class 2 proteins do, their job is to present peptides to T cells. And structural studies have shown, and this has been reviewed by my group and others, that the diabetes susceptible HLA class 2 molecules have specific structural features that allow them to anchor and present autoreactive peptides from islet cell antigens in very specific ways to T cells. And the focal point for much of those structural features is their lack of a aspartic acid position 57 of the beta chain, which opens up pocket 9, so that it has a disrupted salt bridge, and that creates a very large pocket that craves a negative charge. And indeed, many of the most important ligands in the disease, including the insulin B9-23 peptide and the hybrid insulin peptides that are becoming increasingly noted and talked about in the disease, have an acidic residue that binds in this pocket 9, stabilizing the molecule, and creating antigens and neoantigens that are potent mediators of T cell activation, and that are believed to contribute to the disease process. Now, importantly, autoreactive T cells have been shown to be present before disease onset. And again, I could show many different data sets. This particular data set was collected in my lab using HLA class 1 tetramer staining, using a multicolor assay that was first developed in Bart Rope's lab. And the different symbols here show the observed percentages of CD8 positive T cells that are labeled with HLA class 1 tetramers in either established and new onset diabetes for insulin B, preproinsulin, or a pool of IA2 and GAD tetramers. And I'll draw your attention to this elevated bar here with the half-filled circles. So these are new onset individuals that have a very broad insulin B response. But if you look over here at the next group with these open circles here, we see very high frequencies in many cases of PPI-specific T cells in pre-diabetic individuals. And so these T cells are present prior to the onset of disease. And of course, most importantly, and again, I could have chosen many papers and many figures. This paper happens to be out of a paper written by Roberto Maloney back in 2018. And this is just, an active demonstration that autoreactive T cells can kill human beta cells. And so this is a real-time cytotoxicity assay that's being done with this clone D222D. That's a human T cell clone that sees an IGRP peptide. And that T cell clone is targeting ECN90 cells, which is a human beta cell line. And what this figure basically shows is whether you have untreated cells or cytokine treated cells, you can see very potent killing of these target human beta cells by an autoreactive T cell line. And so kind of summing things up, basically T cells are necessary efficient in the animal models. They're genetically linked in human disease. They have been shown to be present in circulation prior to disease. They've been shown to make it to the beta cells, and they've been shown to have the capacity to kill human beta cells, which seems to be a key element of the root cause of the disease. And finally, autoreactive T cells are antigen experienced in individuals prior to disease onset. So this is a single cell data set that we collected from individuals that participated in the teplizumab prevention trial. And so we tetramer labeled and sorted these cells, and then did RNA seq analysis. And I'll point out that basically these cells, when analyzed and by a UMAP analysis, clustered into 5 distinct clusters that include activated T cell sets and naive T cell sets, but then several different effector and progenitor memory cells, along with some exhausted cells. And so, yes, some of these cells are naive, but even here prior to disease, we have activated T cells that seem to be poised to carry out effector functions. And finally, linking these cells, to causality, in my opinion at least, these same autoreactive T cell populations are modulated by therapy. And so again, if we look longitudinally at individuals sampled at baseline or 3 months following treatment with the teplizumab, which was shown to delay the disease, what we see is a moving of T cells, from these effector T cell clusters, like the one in yellow here and like the one in blue here, and diversion of them into this red cluster here where they're exhausted. And so to me, what this supports in a very reasonable fashion, is the classic T cell centric view of the disease. And I agree with Ake that this may be an oversimplification, but I just want to point out that the classic view does hold a lot of water. And so again, this is from a review of our article a number of years ago, but it encapsulates the T cell centric view really nicely. And that is that in individuals that are at genetic risk, there's some still as yet known precipitating event that produces an initial injury, which then mobilizes autoreactive T cells that then kill and impair beta cells, liberating more antigens, recruiting new naive T cells that see different antigens, and this wheel spins. Autoantibodies develop, spread, and finally a person gets type 1 diabetes. And so again, I'm not saying this is the whole story, but I think there is a certain elegance and beauty to the T cell side of the story that certainly shouldn't be thrown out with the bathwater if we find that other cells are important too. And I'll go ahead and stop there.
Great, thank you. Okay, so this is going to be a tag team. Chris will start and I'll do the second half. So let's see. Is that good, Chris?
Perfect. Yeah. Yeah, so I'm skipped the first slide, which is an introduction of the tag team of myself and Ian. And thank you very much, Ellie, for your wonderful overview of the role of T cells in the pathogenesis of type 1 diabetes. And now we would like to introduce another potential player in the pathogenesis of type 1 diabetes, and these are autoantibodies. So thank you also to Ake for the great introduction of islet autoantibodies in type 1 diabetes. And I have been always thrilled to see the strong correlation between islet autoantibodies and type 1 diabetes, which sort of intuitively suggests there may be a causative effect. But then the intracellular nature of the autoantigen itself appears to put such a correlation into question. But when you look at the literature, there are actually a number of autoimmune diseases where the autoantibodies target intracellular targets and have a pathogenic outcomes. And I list just a very few here. Most pertinent for us is stiff person syndrome, which is an autoimmune neurological disease with a very high titers of GAD65 antibodies also in the CNS. And what we were able to show was that in stiff person syndrome, the GAD65 antibodies accumulate in neurons of the cerebellum, where they impair GABAergic neurotransmission, resulting in the characteristic disease symptoms. So what we learn from these examples is that autoantibodies, maybe opposed to regular antibodies, get into cells and do impair function and seem to have a role in pathogenesis. So the question that Ian and I came up with was why does this not happen in type 1 diabetes? So what we did, we initially repeated some of the experiments that Ake described, namely we incubated islets with purified IgG from type 1 diabetes patients, and then looked at insulin secretion. And you see here on the very left, that's a control. On the very right is control IgG isolated from a healthy individual. And then in the middle, we have 2 type 1 diabetes patients, the IgG isolated from them. These are very well characterized patients from the so-called Sweden 10. And what we saw is a modest but reproducible inhibition of glucose-stimulated insulin secretion by both patients. So we were happy that we could reproduce the earlier findings, but we realized that working with the patients serum will be very difficult in identifying the involved mechanisms because, as you know, serum contain a mixture of antibodies to a wide range of targets. So it makes it difficult to hone down on a specific mechanism. That can be, luckily, this can be overcome by the use of monoclonal antibodies. So if you want to go to the next slide, please. So we decided to go and use human monoclonal antibodies specific to GAD65, and we were lucky to have two B cell lines, human B cell lines, specific to GAD65, and cloned these antibodies and characterized them. So this is one of the major slides here. So I'm going to take a little time on them. So as I said, we isolated and characterized these two GAD65 antibodies isolated from a human patient. And in the top left, you see where we labeled the antibodies with 488 with a fluorescent marker and incubated islets with the fluorescently labeled antibodies and observed over time an increase in the signal, indicating that there's an uptake of the signal by the islets. And on the bottom, you see these are dispersed islets, so these are beta cells, and so that's a confocal microscopy of single islets, single beta cells incubated with one of the monoclonal antibodies, and you can see that it shows a very nice staining of the trans-Golgi apparatus. We also did some co-staining with antibodies to the Golgi, and we see that both antibodies appear to stain the same subcellular structure. And as you can also see, it does not stain the nucleus, which in our eyes, and I think we will have some debate on that, indicate that this is indeed intracellular staining. So then we took those two monoclonal antibodies and put them into our glucose-stimulated insulin secretion assay. These are the islets at 3 millimolar glucose, and then we increase it to 20 millimolar glucose, and you get the expected increase in insulin secretion. And what we could see is that one of the monoclonal antibodies inhibited the insulin secretion by 40%, while the other one did not. And this experiment has been repeated several times, and Ian will talk to this at great, will elaborate on this experiment and our petition on that. We were able to publish this in 2022 and continue to work on the project. So if you want to go on to the next slide. So in the meantime, we were lucky to have access to 6, or actually to 4 further human B cell lines specific to GAD65. And we sequenced the monoclonal antibodies, expressed them, and characterized them extensively for their affinity, for epitope specificity, and for inhibition of GAD65 activity. And as you can see, there's a wide range of affinity. Some of them, the B78, that is the one that inhibits the insulin secretion, has a very high affinity down to omega-5, which has a much lower affinity. Some of them inhibit GAD65 enzymatic activities, others do not, and some we didn't get around to test. We have also a human monoclonal antibody to Staphylococcus aureus, which is our negative control. So if you want to go to the next slide. So we took all 6 GAD65 monoclonal antibodies and our control antibody, and again, threw them into our insulin secretion assay. And what we could see was that only B78 inhibited the insulin secretion. We get a little bit inhibition also by DPD, but this one is the one that is most robust inhibition, which we get each time we tested. And B78, if you remember from the previous slide, is also the one with the highest affinity. Whether this is of importance for the mechanism, we do not know yet, but we we're certainly working on finding out the mechanisms. And that's when I hand it over to Ian, because this is a difficult question, which I...
All right, so after we got those initial exciting but very preliminary results that the B78 did decrease insulin secretion, we wanted to ask a number of questions to show that it wasn't a fluke or some kind of toxicity that was unrelated to the actual GAD65 protein. And so there's a natural control with mouse islets that don't express GAD65. So we compared the insulin secretion with rat and human islets and mouse islets, and then showed clearly that the mouse islets don't respond to 3 days culture in the presence of the GAD65 monoclonal antibody that we've been working with. At the same time, we measured GABA secretion. We were expecting that it would inhibit the release of GABA, the antibody. So, and that was borne out again in the rat and the human, and there is a slight decrease. It's not statistically significant, and we think that it parallels the lack of effect with insulin secretion. And finally, we measured ATP at the same time, which again also paralleled. This is ATP content after the 3 days culture. And of course, ATP generation is closely linked to glucose-stimulated insulin secretion. So it made sense that we would look at that, and it certainly offers another window into what the mechanism might be in terms of how the GAD65 antibody is inhibiting insulin secretion. And show just some characterization of the kinetics. And as I say, that previous one was done after 3 days, but we examined insulin secretion after 1, 2, 3, and 6 days of culture with the GAD65 monoclonal antibody, and also looked at the secretory rate of GABA. And it takes about 3 days to become close to what we think is the max. It's only a small increase after 6 days over 3, although we certainly don't know and are eager to find out what happens at much longer periods of time. But certainly the mechanism seems operational after 3 days. And I think it's worth noting that the insulin secretion doesn't start to go down until after the GABA secretory rate has already gone down, which, and I'll talk more about this later, but it does potentially suggest that actually the GABA, the release of GABA and the enzymatic activity of production of GABA may not be actually what's mediating the effect of the antibody. So as I say, I'll talk about that later. But using these conditions, the 3 days, we needed to know that this was operational in vivo, and we used a rat model. And it was certainly convenient that the antibody did the same thing in the rat islets as it did in the human islets. And given the history of isolated islets to very well respond similarly to in vivo secretory responses, we predicted that we would see the same type of inhibition of insulin secretion after 3 days. So we used the same culture, same conditions in vivo as we did in the culture of isolated islets, and we did an IP injection of GAD65 monoclonal antibody at the amount that would, assuming 20 mils of blood, that would give us the same concentration in the blood that we've been using in the in vitro experiments, and also that we left it for the same time. We did 12 rats of each, where one got the GAD65 monoclonal antibody. The other one, as Chris said, was our control for the staph monoclonal antibody. And in general, of all the experiments we did with isolated islets and in vivo, we never saw any effect of the control monoclonal antibody. So the experimental design was during these 3 days, is each day we took a sample of blood, and the main data that I'm going to talk about for these first 3 days is that GAD65 monoclonal antibody went up in the blood within an hour of injection, and it stayed elevated without really any sign of clearance up until day 3. So that was constant. And the important data that we got on this day was measurement of insulin before and then 15 minutes after injection, IP injection of glucose. So the experiment is 15 minutes after an IP injection of glucose and 3 days after the IP injection of the GAD65. And so just a simple summary of the data we got, that here's our control, the amount of plasma, insulin in the plasma at 15 minutes after the glucose, and is reduced by 65% or so in the animals that received the injection of the GAD65 antibody. So this was very reassuring and exciting for us. So just at this point, it looks like we have data that suggests that the GAD65 gets into beta cells. And I think this is the first time that we've seen in isolated islets that GAD65 suppressed insulin secretion, did it in a time and concentration dependent fashion. I didn't show the concentration dependency, but it's about 1/5000th of what the total IgG is that we begin to see effects. So it looks very plausible that it's not high levels that we're looking at, and only happens in islets that contain GAD65. So there's an absolute requirement that it's antigen. And finally, we've recapitulated the effects of GAD65 antibody in vivo. Okay, so now to go on and look at the mechanism, we have, let's see, this is summary. So this is, let me go on. I got ahead of myself. No, actually, okay. I'm going to summarize. I'm not going to show the data in the interest of time. We did already see that GAD65 antibody inhibits GABA secretion. And I showed that islet metabolism is also inhibited in terms of ATP, but we've also done experiments showing that there's a similar decrease in the oxygen consumption response and NADH response to glucose. And so we certainly don't yet understand the mechanism, but given that GABA in general and the experimental data that I haven't shown that has only a very weak effect on insulin secretion, it seems much more likely that it's associated, the mechanism is associated with a decrease in generation of ATP, although we still have a lot of work to do to understand how these are all related. And then as Chris showed, this is actually somewhat, at least at this point, with only 6, it may be a very rare event out of all the antibodies that are present in an antibody-positive patient. There may be a very small number of actually deleterious antibodies that can affect insulin secretion. Okay, so we've actually submitted this paper now and hope to get reviews back. So I'm going to make a single conclusion, just lest anybody think we're over-interpreting our data, and that we've just shown that there's this one single GAD65 idiotype that reversibly impaired insulin secretion. And we want to be careful that this demonstrates the plausibility of a direct pathogenic effect of the antibody in the development of type 1 diabetes. But we certainly have a long way to go. And there's many questions remain to be answered before contribution of antibodies to type 1 diabetes has been demonstrated. And I have a list, but I'm going to hold off on the list because Sally Clark is going to present next and did such a wonderful job on telling us what we need to do next. And I'll hold off on that until she presents those. And I just want to finish with some future work that we've actually written in grants and plan to do. One of the most important one is extending the exposure of islets in our in vivo experiments, where we did it for 3 days, and we really want to see over 1 to 12 weeks whether this decrease in insulin secretion may be further developed into actual loss of beta cell mass or even immune response by cells. We think this is actually a very important model. We're glad that it works in the rat islets because we can use not only the isolated islet, but the in vivo model to screen for the ability to prevent antibody-induced loss of insulin secretory capacity. The second, we clearly need to look at the mechanisms, which we don't have a good handle yet. And this is also, we think, very important that we want to identify these mechanisms, and I'll talk about this a little in the last slide, that may be more amenable if we can block one of these steps over the cellular responses that are very complicated. And I do want to talk about the last future work in a little more detail. There's a new method called the beacon instrumentation. And in the past, it took Chris many months to generate a monoclonal of all the steps involved. And so this new beacon analysis can generate monoclonal antibodies in a single day. It has 40,000 wells they call pens, and you can put one B cell into each of those wells and generate monoclonals and then screen those for the targets. So we're very excited about using that to characterize effects and mechanisms of actions, not only of individual, but as Ake pointed out, the importance of looking at the combination of autoantibodies to see if they have synergistic effects. And also, one of the major questions we need to answer is what is the prevalence of harmful monoclonal antibody idiotypes in patients, to see whether we have a fluke and we've identified the only one and only, or whether each patient is going to have a deleterious set of antibodies. And I just want to finish with one of the major reasons I'm excited about this whole project, in my mind, in terms of what Eddie said. I certainly have not a deep understanding of these processes, but what we're hoping for is within this landscape of immune cells, whether it involves B cells, T cells, and other immune cells, that our premise would be that GAD65 antibody and other antibodies play a necessary step in the loss of beta cell mass and function, so that it may not be the whole story, but if it's essential, then we could block those steps, possibly even the uptake of GAD65 antibody and other antibodies, or the intracellular steps, and that would be enough to delay the onset of the disease.
Okay, that's great. I think we're at 12:45 now, so I didn't know if Dr. Kent wanted to weigh in on her section.
Absolutely.
There are several questions that we probably want to get to after she finishes speaking.
Right. We may not answer as in depth as time permits. So,
okay, so thank you. I want to just bring up a couple of points. I won't take too long. I think I'm going to advocate for you looking at whole patient sera on the next slide. But when you do, you need to have a very clear idea of what patient cohort you're looking at. I believe Laura Jacobson is on the call, and she can weigh in on some of these. The occurrence of GATA positivity is different with the age of the donor or the age of the patient when they develop. And so it's really important to know what cohort you're getting this from. I don't know what patients these antibodies were identified from and what stage of the disease. That's really important to know. I think I couldn't find a good reference for this, but I also have heard incidentally over the years that many people are single GATA positive without developing the disease. So you have to worry about that sort of thing too. Also, you've mentioned this a little bit, you've got a spotlight effect going on here. You've got anti-GAD antibodies, so you're claiming there's a harmful effect of the anti-GAD response. And you do have to look at other autoantibody specificities, which you just looked at or just mentioned. So I really think you need to do that. Be aware that you need to know what cohort of antibody patients you're looking at for autoantibodies to GAD. Next slide, please. So I came up with a variety of questions that I'd want to have answered to understand if this is really something we need to pay attention to. I believe a number of the people in the audience will have questions too, that you will please bring those up. So you're mostly using a rat islet system, and you did say yes, that this rat GAD65 is homologous to human GAD65, which is important. I wanted to know about the physiochemical differences between the two anti-human GAD antibodies. And you said there's a difference in affinity and ability to maximally inhibit GAD65. bind to different epitopes, you need to mention the epitopes and what they look like. They're both conformational, I believe. You need to show those at some point. I know that's old data, but bring it up again. What constitutes a potentially harmful anti-GAD antibody versus a benign anti-GAD antibody? I think you need to do a lot more here to show that what's going on with the inhibition of the insulin secretion and GABA release and generation of ATP. We need, as you mentioned, we need to know the mechanism. We need to know all the steps and how that is deleterious, or is it not really a deleterious thing that's going on? The other huge thing you have to answer, how frequent are the potentially harmful anti-GAD antibodies? And you're going to have to screen patient sera and figure out a way to isolate the anti-GAD response from the sera, which I think there's probably better ways to do now. And just be aware of the cohort you're using. Will any patient sera with a GAD titer have the same effect? And you answered no. How do you know the concentrations of the potentially harmful GAD65 antibodies are physiologic? I'm not really buying your answer here. You don't know the answer. It doesn't matter if it's 1 to 5,000 or 1 to 20,000. You don't know if it's physiologic or not. How do these potentially harmful antibodies arise? We don't know. That may be something you need to think about quite a bit. And then I asked, what happens to the beta cells if you add purified GAD65 antibodies from GAD-positive donors to live pancreas slices from a control donor? And you answered, well, you're going to have to figure out a way to purify those from patient sera. Slices can last for up to 10 days. People have been doing that on a regular basis. So think about that and really think about using human islets and human pancreas slices. Next slide, please. So I want to know what the mechanism of the anti-GAD internalization is. I think that's really important. I asked if antibodies are in vesicles possibly designated for degradation. Do islets have many FC receptors? You said don't. I'm not completely convinced by your immunofluorescence. I know Sarah Richardson is on the call, and we've had to do a lot of tricky things to show that things are on the surface or internal, even if it's not staining the nucleus area. I think you really need to nail that down in a different ways. And if you'd like, we can talk about that. Have you measured specific alterations in GABA release? You said yes. It's inhibited in parallel with ATP and insulin secretion release. The mechanism, this is really got to be nailed down, how this is happening, how the antibody is actually doing that. So you're going to be having to address that. I think that might be my last slide. Yes. Yeah. And I definitely want feedback from the audience of other things they need to do to show that this is a real phenomenon.
There is a series. Yeah. There are a series of comments and questions in the chat. Would you like to take those, or would you like to have responses? People unmute and weigh in with Sally Kent's.
I think we should go to the chat. Sally and I and Oka, we can discuss with her later. I want to hear new input from some people.
Okay. So from Bart Roop, I'm confused. If mouse beta cells don't have GAD65, how can GADA impair insulin secretion? By the way, in an excessive antibody concentration, ...
I think we did answer this already in the chat a little bit. The inhibition that we showed was only in human and rat islets. Particularly, we could not show it in mouse islets because they don't have GAD65. So I think
Chris, is that also in the in vivo experiments? I was maybe a little confused about that.
Yeah, we looked only at rats. We didn't look at mice because they don't have GAD65. So there must be other autoantibodies that have a function in, or if it's on autoantibodies, these must be other islet autoantibodies.
But I noted 0.8 milligram per mil. That has never been seen in not even in stiff person syndrome. Is that correct?
Oh, that's in a way I shouldn't have written it that way. That's what we added, but once it gets in the blood, it's then diluted to 0.04 mg per mil. So that was just the solution that we added in terms of what we injected. But you're right, that would be very high.
But I also noticed in some of the first blocking experiments, it was 250 microgram per mil concentration of antibody, which is really high.
I don't know. I don't know which one you're referring to. We never did that.
The first ones that Chris cited in the introduction.
I could get the slides, but
in the interest of time, let's move on to the next question while you search for that. From Sarah Richardson, interesting work, Ian and Chris. Do you know if the GAD65 autoantibody that inhibits insulin secretion targets the truncated GAD in autoantibody assays? Do you think it could be triggering beta cells dedifferentiation or transdifferentiation?
So yeah, this is a great question. And we certainly have to look into the dedifferentiation of beta cells. It's something that we will be able to look at in the in vivo experiments. As to whether it targets the truncated version of GAD65, we have not tested that yet. But based on the epitopes that they recognize, it should bind to the truncated version as well. But we have to do the experiment for sure.
A follow-up question from Sarah Richardson is, could you try labeling an antibody with a fluorochrome and allow internalization and then quench the fluorochrome outside the cells? If it gets inside the cells, you should see fluorescence still inside. There's a system to test this, but we need to look into it. Remember it.
Yeah, that's great, and that's exactly what we would like to get from Sarah and also from Sadie, an experiment, how to show that they're really inside of the cells. When we did we labeled the antibodies with fluorochrome, but we didn't do the quenching experiment. So thank you very much for that. We really look forward to doing that experiment.
Okay, we're at 12:55, so we have 5 minutes left for questions or comments. Do you want to? Oh, here's another comment from Eddie James. In support of Ian and Chris paradigm, there are a few studies that support the necessity of beta cells in the NOD model, and anti-CD20 did have therapeutic benefits, although some have proposed that this reflects on essential APC function for B cells. I don't know if you want to unmute yourself, Eddie, and you can discuss that live.
Oh, well, I mean, this is, you know, let's let the other questions, you know, be asked first.
Sure.
We want me to get back to the slide that I try to identify the high concentrations.
Sure, why not? You can, if you found it, you can share it.
Yeah. So is this the one you were talking about?
Yes.
Oh, okay.
Okay, Chris, is this a concentration that's high?
That is, well, that's the total IgG. That's not total GAD antibodies. So we purified total IgG from the patient.
Okay, and would you mind to move on, Ian, to your in vivo studies, just to get my mistake out of the way?
Yes. This one. Yeah, so...
Okay, yeah, that looks more like a rat, you're right. It seemed like a mouse at first, but...
No, it's a tail that gives it away.
No, you're absolutely right. I should put rat somewhere here. The artistry is not so good. Okay.
Thank you.
Yes.
Let me raise a question. My name is Yehuda Schenfeld, and there are several people from our group here in the audience. We are not expert in the issue of anti-GAD and insulin, but we have quite experience with idiotypes and pathogenic idiotypes and dominant idiotypes, etc. So first of all, you have only one monoclonal, the B78, which was found to be, so to speak, pathogenic. So the suggestion is to see if it is important, and you have to generate anti-idiotypes to the B78, and then to screen patients who developed insulin-dependent, the type 1 diabetes, and to see if this antibody is prevalent. So it's an association. It's not causation, but then you can say, wow, we found a pathogenic idiotype, which is dominant. By the way, it can be used eventually as a diagnostic tool to differentiate between those anti-GAD which are non-pathogenic and maybe just epiphenomenon, and from those who are pathogenic. The other issue that I wanted to ask you, but forgive me, it's an ignorant question. I have Dr. Efrat Wertheimer with us. She is more experienced with the insulin receptor. Is it possible that your monoclonal, the B78 or any pathogenic gut antibody, binds to the insulin receptor by cross reaction, by having similar epitopes, having gut 65 on the top, and so forth? And last but not least is, if you want to show that the autoantibody is pathogenic, you have to show that if you can neutralize it, you may prevent the reduction in the level of the glucose or the insulin response. And you can do it by two ways. Either use a compound, the IVIG, intravenous gamma globulin, which you may expect that it contains natural anti-idiotypic antibodies, or if you will generate the anti-idiotypic antibodies in another species, you can use this to show that when you neutralize it, you can prevent the pathogenicity of the B78. And that's what we offer to collaborate eventually. I don't know, Efrat, would you like to say something?
I'm to tell the truth, I'm in a learning phase of looking at all this complexity. There are evidence that really the GAD65 interact, as Professor Schoenfeld said, interacts with the insulin receptor, and that could affect really the well-being and the activity of the islet cells and the secretion of cells. I was looking more at it, looking at diabetes type 2 and insulin resistance that can be caused also by all these interactions. And this is all very interesting, and we should be looking at it as well.
That sounds good. Perhaps there's a chance for a potential collaboration here. And thanks for sharing. I have one more question. We're at 101, but I'll have one question from Matthias van Hereth. In the chat, I wonder whether certain autoantibodies could serve as an attractant of T cells to islets. This would have to be done in human settings, and I like the slice idea. I don't think the animal models would teach us as much or on that question. 10 days of slice survival might actually be enough? ?
I mean, I'm happy to go ahead, Chris.
I mean, it's certainly possible that autoantibodies could attract T cells to the beta cells. That's a very attractive idea. Cannot say much about the slices, sorry.
Well, actually, we've started getting, actually this week we're doing a rat pancreatic slice in order to get the methods up and running, and Julia Panzer at City of Hope is going to start sending us, when they get them, the human pancreatic slices. I didn't know they could last for that long, so I'm actually very pleased. And we can definitely work with that tissue in response. So this is a great suggestion. We're definitely going to go forward with that.
So
yeah, and maybe I think, Yehuda, good to see you after a long time. This is Matthias. I mean, maybe the anti-idiotype idea is a good one, because we revisited this old plasmapheresis study that Johnny Ludwigson and I think Hubert Korb and Ake, you had been involved in this. And they were not fully conclusive, but they are hard to do. This anti-idiotype idea might be much easier to do than plasmapheresis in humans, which you would have to do for God knows how long. And that's complicated, right? It's a very interesting area.
Chris is the expert in this, so Chris.
You are right, Matthias. Yet, I think that you have a stool here because you have a golden marker, and this is the B78. Luckily enough, when we were involved in the same questions that were raised here regarding lupus, and we had monoclonal anti-DNA antibodies, and the question was raised whether it's only B cells. As you know, anti-DNA per se, when even you passively transfer it to models of mice, you don't induce lupus. By the way, we were able to induce lupus, which we published in PNAS, by idiotypic stimulation and raising anti-idiotype and anti-idiotype, and then the mice developed what we referred the first induced model of lupus. So here you have a marker, the B78. So you can generate the anti-idiotype here very easily if you have enough from the monoclonal antibody. Now, regarding the question, what is the major pathogenic function here, the cells or the antibodies? And it might be a combined action. One can be that the autoantibodies are the first noxious agent, and then they do some morphological changes, and they stimulate the cellular reaction against the islet cells. And
Exactly.
And the contrary, it can be that the cells penetrate at first because of genetic reason, because an autoantigen, etc. And they exposed, for instance, the GAD65 on the cells and generated antibodies. So the antibodies are epiphenomena. And last but not least, whenever they are generated, the cells and the antibodies, they can react combinedly to generate the complete destruction of the islet cell. This is just hypothesis.
From the chat, we have A. James, it's certainly reasonable to think that if beta cell function is compromised, that would contribute to ER stress and trigger the wide variety of neoepitope pathways that my group and others have been studying. Well, I mean, we're at 106 right now. This has been a very fruitful discussion. And I think people have a lot of takeaway to consider and think about and perhaps connect offline.
So I would like to thank on behalf of my group that you have invited us. You opened a new field for us, even though the marker was Chris Hampe, who presented a wonderful presentation, our Friday's Mosaic of Autoimmunity presentation. But we are open for collaboration as well as whatever discussion in this field, and definitely in the next autoimmunity meeting, you will have a major role in the question, is it the cell which are destructive, or are there the autoantibodies? In 1 month, in March, we have controversy in medicine. Unfortunately, in an ugly city of Venice in Italy, but unfortunately, the program is already settled down, and I'm not sure that I will be able to present what has been done here beautifully by all the speakers as an issue. But if there will be a slot, we will be in touch. So thank you very much for exposing us to this wonderful and attractive subject.
Thanks, Yoda.
Yeah.
Thanks to everyone who attended, as well as the speakers for putting forth their eloquent arguments, research, data, and commentary. We appreciate everyone's attendance. This will be recorded and held on the Sugar Science website, and you can access it at any time in a few days. So thanks again, and we look forward to the next series of experiments to come out surrounding this work. Have a great rest of your day, everyone.